Hi, good morning, everyone. I hope you all had a restful weekend. We're going to go ahead and get started with lecture 2, which is blood and the lymphatic system. It's kind of like a continuation of what we've learned last time. Last week, we learned about how blood circulates within the body, where you pick up oxygen, where you deposit them, and so on and so forth. Today, we're going to take a closer look at what is exactly inside the blood and the various components and their biological functions, and we're also going to look at the genetics of blood types. And at the end of the lecture, we will talk a little bit about the lymphatic system, which is essentially part of your immune system. The two kind of work together, and then it will transition nicely into next week's lecture, which is entirely on the immune system. So with that said, let's get started. Now, I saw only 7 or 8 of you have accessed the PowerPoint slides, so please make sure that you do have a copy of the PowerPoints when we go through the lecture. That way you can take notes and follow along. Okay. So blood, right? There are approximately 5 liters of blood in an average person. That's 70 kilograms. Of course, the heavier you are, you're probably gonna have a little bit more blood, but on average, there is about 5 liters of it. And a majority of these blood will be going through the kidneys continuously, right? As the organ is filtering them, right? As much as 1.2 liters of blood goes through your kidneys every minute, and its job is to filter it, take away the waste product, and put it in the urine. So what exactly is blood? The best description for blood is that it is a protein-rich fluid with cellular components in it. As we will learn, there are actually hundreds and thousands of, maybe not thousands, but like hundreds, okay, hundreds of proteins that are found within the blood. And all these proteins, they serve a specific role, but in general, the protein is going to add to the osmotic pressure that we talked about and help retain the fluid within the blood. So it's not going to leak all over the place at all. Give me 1 second. Okay. Now, the other part of blood is that there are cellular components in it. You probably know that there are red cells in it, there are white cells, and then there are platelets as well. There are the 3 major components of cellular of cells inside the blood, and we're going to take a closer look at each one of them. And so if you take blood from a person and you spin it down using a centrifuge. This is a diagram of something called a centrifuge. It spin fluid at really high speed, and that causes the heavier stuff to sink to the bottom of the test tube. Okay, so if you take blood and you spin it down in a centrifuge, you will see that it separates into a clear yellowish fluid at the top. That's what we call the plasma. Okay, so the plasma makes up approximately 55% of all of your blood. Okay, the plasma is mostly made up of water, and then we have some dissolved proteins and minerals and ions in it. The lower portion is what we call the formed elements. Okay, formed elements consist of all the cellular components. Okay, so the red cell that we talk about, the white cells, and the platelets. And you can see the red cells are the ones that are the most abundant. Right? You have a lot of them. You don't have a lot of white cells and platelets. The white cells and the platelets are lighter compared to the red cells, and so they float on top. Right? So you have your plasma, you have your red cells at the bottom, and in between, you have your white cells and platelets. Okay, that forms what we call the Buffy coat. Now, you might not recognize some of these words now, but we will talk about it later. Okay, leukocytes is just a fancy word for white cells. Erythrocytes is basically red blood cells. Okay, so we'll look at those words later. For now, what I want you to understand is when you spin the blood down, you will separate the liquid portion, which we call the plasma. That's 55% of your total blood volume. And then you would have your cellular component, which makes up 45% of your total blood volume. And all the cellular components collectively, the red, the white, and the platelets, we call that the formed elements. Here is what it actually looks like when you do spin the blood down, right? So this over here, that is the plasma. Okay, and down here, maybe I should include this little portion here as well. That's the formed elements. Okay, and this stuff here, the really dark. It looks like black here, but it's just a really dark red. Okay, that is your RBC, red cells, and then you have this not very clear in this picture, but it's kind of like having a little bit of like a cotton. Okay, there it's kind of like cloudy, fluffy, and that's where the white cells and the platelets are going to be. All right. So we're gonna take a closer look at these components and see what's in them and what they do. Plasma. Right, so plasma is a fluid portion, as I mentioned earlier, and it's mostly water. Okay, mostly water. There are other stuff that are dissolved in it, ions. Right? So we have sodium, we have chloride. Anyone know other ions that are important in the blood? Right, we collectively call them electrolytes, right? So what are some other important electrolytes besides sodium and chloride? Does anybody have any?
Potassium.
Yep, potassium, that is definitely one, right? Maybe just one more. Just want to see if anyone else know. When you drink your sport drinks or Gatorade and whatnot, what exactly is in there?
Would there be magnesium?
Yeah, there's going to be a little bit of magnesium as well. Very good. Okay, so all these things are going to be dissolved in your plasma, right? There are various nutrients such as glucose, right? You will have a little bit of amino acids, okay? Also waste products, right? Waste products like we talk about carbon dioxide, we talk about, maybe we haven't, okay, but things like urea, that's going to end up in your urine, and that's a byproduct of breaking down proteins. Okay, so it's not all just like useful stuff in the blood. There are some like waste that we need to get rid of as well. That's why we need the kidneys. That's why we need the liver to filter the blood. And tons of proteins. Okay, so many proteins. We're just gonna look at a few of them, right? And that's what plasma looks like. All right. So I want to tell you something because we can talk about blood transfusion later on. In movies, you might have seen a situation like this happening. Okay, like you have one person, okay, and then you have another person. Okay. Usually you might see this in like war movie or something, okay? This person is bleeding out. So what you see in the movie is they take a line and then they connect it directly to the other person. And then they do the blood transfusion. Have you seen something like that? Right? You might have, right? Okay. What do you think about this concept? Do you think it's safe to do that or not safe? Like is this like a medically sound thing to do? Let's do a poll. What do you think? Like, do you think this is safe? Choose A. If you think this is not safe, choose B. Maybe it's the way I phrased the question, right? Like, oh, if he's asking us is probably not safe, right? The truth is, if you do this, there's a very, very high chance you're gonna end up harming the person, okay? If not kill them outright, okay? In real blood transfusion, okay, we do not transfuse whole blood. You take the blood out and you actually separate it into RBC, the red cells, and the plasma. Okay, and we kind of leave out the white cells in this process, and then you will transfuse the plasma separately from the red cells, because the rule for transfusing red cells are actually different from the rules for transfusing plasma. Okay, so the rules are actually opposite to each other, and therefore we cannot transfuse whole blood. Okay, and you can see in this picture, they actually store the plasma separately, right, from the red cells. You would extract the plasma after you spin it down, and then you have a bag of plasma, and this plasma is from a person who is B positive. You can see B positive. All right, so I just want you to remember that. Okay? In actual blood transfusion, we do not do the whole blood. We separate it, and the rules are different, and we'll learn the rules later on. So formed elements, right? That's the 45% of the blood that we saw, and it consists of, again, the three types of cells, including the red cells, which makes up a majority of the formed elements. So that really packed dark red portion, it has another name. We call it the hematocrit. So the hematocrit is only the red cell portion. It does not include the white cell. It does not include the platelets. And the function of the hematocrit is basically to deliver oxygen. That's what red cells are for, right? And transport oxygen throughout the body. So when people say blood transport oxygen, it's not really blood as in like the whole blood, okay? It's just the red cell portion that does that for you. There are also white blood cells, which you probably know help defend the body against pathogens. So pathogens are living things that can cause us to become sick. So things like bacteria, okay, virus. Are there other living things that you might know that could cause the body to become ill other than, bacteria and virus? Does anybody have a
Parasite?
parasites? Yeah. Do you have a specific example of a parasite, maybe? Just wondering.
Um, maybe like a tapeworm?
Yeah. Sure. Tapeworm, right? Yeah. Malaria. It doesn't have to be so big, right? It could be microscopic malaria, right? And one more. Anyone else? Bacteria, virus, parasites. What other pathogens are there? Okay, CSF fungi. That's right, fungi. So if you have like a fungal infection, that's considered as a pathogen. All right. So it has to be a living organism, and the white cell's job is to defend the body against these things. Right. So not everything that makes you sick is a pathogen, right? Okay. Like if you ingested some chemical that's poisonous, right? That's not a pathogen. A person can have cancer. That's not a pathogen. Cancer arises from your own cells, right? So that's not considered as a foreign substance. Okay, so these are foreign living organisms that makes us ill. Platelets, finally, is going to help you stop the bleeding. So if you have a cut and the bleeding stops, you form a scab, that is the job of platelets. All right, so there you have it. Those are your formed elements. Remember, a majority of it is the hematocrit. The white cells and the platelets is like the Buffy code, which is less than 1% of your total blood volume. Let's take a closer look at the plasma portion. We also have, I said we have a lot of proteins in the plasma, and the most abundant one, the one that you have a lot of, that's what abundant means, is something called albumin. Albumin, all right? And it's made in the liver. In case you don't know where your liver is on the top right quadrant of your abdomen, of your abdominal cavity. Okay, so this is your diaphragm. And then just underneath the diaphragm, that's where you will find your liver on the right side, top right. Okay, just a brief note about the liver. You can lose like 1/3 of your liver and it will grow back. Right? So that's why a lot of people donate their liver for liver transplant. They cut off 1/3 of it, and then after some time, it grows back. It's remarkable in its regenerative capability. Okay, but the liver, amongst other things, is responsible for making a lot of the plasma protein. Okay? And the key one that it makes is something called albumin. Okay, now albumin doesn't have like a specific function in terms of doing something on its own. But the albumin, since you have a lot of it, it is the main protein that helps you maintain the osmotic pressure. Okay, so we talked about this last time, right? If you think about the capillaries and you have all these dots on the inside, right? And then you have some dots on the outside. So a lot of these dots are actually albumin, and that helps you retain the fluid in your blood vessels. I'm sure you have seen pictures like these, or sometimes even in posters for like World Vision and whatnot, that shows you kids who are starving from less fortunate parts of the world. And one of the hallmarks of these pictures is that all these kids, even though they are starving, you will see that they have a big tummy. Okay, and you may wonder why. How come they have a big swollen tummy? And the reason is because when you are malnutrition, right, you're not going to have enough dietary protein. Okay, because you're not eating a lot of meat, for example, right? Like these kids might survive on grains alone, right? A lot of times maybe sugar, but they don't have a lot of meats or sources of protein, high quality protein. And as a result, they don't have the raw material to build their own proteins. Okay, we learned this last semester, right? The macromolecules. You take your proteins, your body breaks it down into amino acids, and then you use those amino acids to build proteins that you need, right? But if you don't have enough raw material, then you end up not making proteins that you need. You're not making things like albumin. So for these kids, they actually would have a lower concentration, lower plasma concentration in their blood, right? So let me try to erase some of these dots, right? Because of the low albumin. And so the osmotic pressure is now lower, right? It's not as big as before. And the blood pressure is the dominating force that's pushing fluid out, right? So you have a lot of fluid that are leaking into the surrounding cells. Okay? Those fluids, some of them return to blood, but majority of them stays in the tissue, causing swelling. Now, how come it is only the abdomen that's swelling and not the arms and not the legs? Well, we talked about how when we move our muscles, the muscle serves as a kind of pump to return these fluids back into circulation, right? Okay, but because there's not like a lot of movement in the abdomens, right? We don't really use our abdominal muscle all that much, right? So they tend to pool there, and as a result, cause these swelling. Okay, so like a big swollen tummy is actually caused by low protein in the diet, resulting in things like low albumin. Okay, hope that makes sense. Now, there are other types of plasma proteins. Some are important for immune response. Others are important for coagulation process. Coagulation is to stop bleeding, clotting of blood. Right, so we have two over here that we will examine closer later on. One is called fibrinogen, and the other is prothrombin. And both of these are necessary for blood clotting. You probably have heard of antibodies by now. Okay, these are Y-shaped proteins, and we need them for immune defense. Okay, you can make new antibodies every time you encounter a new pathogen. And you have some antibodies that are circulating in your system at all times, antibodies for things that you have fought off previously. Okay. So if you have had COVID and you recovered, now you have some antibodies against the virus. If you had a vaccination, right, for the COVID, you will also have some antibodies in your system. So all these are found in the plasma, okay, of the blood. So where do all the cells come from, right? In the blood, right? The red cells, the white cells, and the platelets, where do they come from? They all came from stem cells in bone marrow. Okay, so if you take a look at like a long bone, for example, this is like a bone, it's like a bone in your leg, right? Your femur. The inside is a hollow space. Okay, and that's where you will find your bone marrow. Right? People eat this stuff, right? Like not the human ones, obviously, but like cows and pork and whatnot, right? It's actually people like to bake them and eat them, for example. So bone marrow contains stem cells, and these stem cells will continuously create new blood cells, okay, in a process called hematopoiesis. Hematopoiesis. This is the creation of new blood cells. This is a continuous process that keeps on happening as long as we're alive, and we would get it from these active bone marrow. Now, in kids, during fetal development and young kids, a lot of the bone marrows are active and they keep on churning out new blood cells. But in adults, only certain bone marrows are active. Okay, a lot of it is actually get converted as we age from red bone marrow to yellow bone marrow. Okay, so yellow bone marrow is primarily just for fat storage. It's the red one that actively keeps on making more blood cells. Okay, so we can see here we have a stem cell, and then under the different type of stimulation, right, with growth factors, with perhaps hormones, they will choose a particular path, right? So they actually become these things called erythroblasts and then reticulocytes, and ultimately it becomes the red cell. Okay, and then if you want to become a platelet, you're gonna do a different paths. And there are actually many different types of white cells. And again, depending on the type of stimulation the cells receives, it will become different types of white cells. Now, because before you get too concerned about this slide, you don't have to memorize it, okay? What you have to know is that hematopoiesis is the creation of new blood cells, and that happens in the bone marrow, where you have stem cells for these things. Are there any questions before I move on? Okay, so let's take a closer look at the cellular components, starting with our red cell. That's what red cells look like. Again, you have a lot of it. They make up your hematocrit. And the fancy word for red blood cell is erythrocytes. Okay, erythro means red, and cyte means cells. So this is erythrocytes. Okay, think of red blood cells as one of those jelly-filled donuts. I don't know if you ever had one. It's kind of like a disc, and then you pinch in the middle on both sides, and that creates this little indentation. So that's what a healthy red cell look like. The red cells don't have a nucleus, right? You remember learning about eukaryotic cells and prokaryotic cells last semester. Even though we are eukaryotic cells, right? The red cells, which is part of us, do not actually have a nucleus. Without a nucleus, it allows the cells to have that indentation in the middle. If you look at it from the side, right, it's indented on both sides, right? So that creates what we call like a biconcave shape. Concave means is caved in, and then it's caved in on both sides. That's why it's biconcave. And that actually increases the surface area for gas exchange. Okay, so oxygen can go in and come out easier when you have a larger surface area dedicated for that. The red cells contains hemoglobin, okay, which is a protein that actually helps transport the oxygen. And over here, you can see a picture of hemoglobin. It has two beta chains and two alpha chains, right? Last semester, we learned about the quaternary structure of a protein, right? So that's what hemoglobin is. It's not made up of just one polypeptide. There are 4 polypeptides in it, and within each of these polypeptides in the center, there's something called a heme group. Okay, the heme group contains a metal. Anybody know what metal it is in the heme group?
Iron?
Iron, that's right. Resounding iron. Very good. So just gonna put iron in case you don't know what Fe is. So that iron allows you to carry the oxygen and you transport it throughout the body. Now, if you have a deficiency or defect in the hemoglobin or red blood cells, that will lead to anemia. Okay, so anemia basically is a group of conditions where you are not able to efficiently distribute oxygen in the body. So there are many different types of anemia depending on the cause, right? So deficiency in hemoglobin means you don't make enough of it. Okay, defect in hemoglobin means maybe you have a mutation that causes the hemoglobin to change its shape and therefore not function properly. But it's also possible that you don't make enough red cells or you lost a lot of it. Okay, so like someone has a excessive blood loss that could lead to temporary anemia as well. So, many different types of anemia, but I'm sure you know one of them, right? Because you probably learned about it last semester. Anybody want to tell me which one I'm talking about? Which type of anemia have you learned last semester or have you heard of?
Sickle cell anemia?
That's right. Thank you, Leah. That is the sickle cell anemia. If you don't know what that is, that's okay. I will refresh your memory in a bit. But let's move on for now. Oh, sorry. I'm going to refresh your memory right now. I just don't remember what slides comes up. It's as much of a surprise to me as it is to you. Okay, so sickle cell anemia is caused by a missense mutation. This is the sequence of the wild type, the normal hemoglobin. And if there is a mutation over here, right, that causes it to change from a thymine to an adenine, then it codes for a different amino acid. And that single point mutation is enough to cause your red cell to change from the normal shape to this crescent sickle shape. Okay, and these red cells, they do not transport oxygen very well because they get stuck in the capillaries. Okay, and sometimes when they do get stuck in the capillaries, it causes quite a bit of pain. And so, people with sickle cell anemia, they would be tired because of not getting enough oxygen, and shortness of breath, that kind of stuff when they physically exert themselves. So that's one type of anemia. Now, sickle cell anemia is a recessive condition, and you should know what that means again from the genetics unit last semester. So if someone is big S, then they are going to be normal. Okay, and if they are small s, then they are going to have the sickle cell anemia. Now, I know if you were with me, you learned the genetics, right? But the rest of you also learned the genetics. Do you guys understand what I mean? Like when I say big S, small s, just quick poll. Because we're gonna be talking about this quite a bit later on today. Do you guys understand? Big S, small s? A for yes, B for no. Okay, good. Wonderful. All right. So what about big S, small s? Do you think these people have sickle cell anemia or they don't have sickle cell anemia? What do you think, huh?
They're carriers.
They are carriers. So do they have sickle cell anemia or not?
They're normal, but they carry the gene.
That's right. So they're for the most part normal. They don't have sickle cell anemia, even though they have the defective gene. Okay. Now here's the thing. Here's the thing that you might not know. Okay. Malaria malaria is a parasitic infection, right? Spread by mosquitoes. And this heat map shows you, places in the world where malaria is very prevalent. Okay, so a lot of the Southern African countries, a lot of malaria. Southern Asia country also have a lot of malaria. And interestingly, when you look at the sickle cell anemia gene in these regions, for people who lives in these parts of the world, a majority of them is actually big S, okay? Instead of, the normal that you would expect. And this is the reason. It turns out if a person has sickle cell anemia, or if they even have one copy of the mutated gene, they are immune to malaria. They don't actually catch it, okay? Because in order for the parasite to replicate, they need to infect your red cells. And if the red cell is deformed in shape, then the parasites can get in and replicate. And so for people who live in places where there's a lot of malaria, the heterozygous condition is actually the most protective, okay? Because the small s protects you from malaria, and the big S makes sure that you don't actually have the symptoms of anemia. Okay, so it's the most popular genotype in those places. Okay, just thought you might want to know that. Moving on, we have the more information about red cells. We know the red cells are formed from the red bone marrow stem cell. Right? The process is called hematopoiesis, right? Okay, and as the red cells level decline, because they might die off, and you need more of them, or sometimes if you move to a higher altitude, like places where air is thinner, for example, then that triggers your body to produce a hormone called erythropoietin. You can just call it EPO. Okay, so the EPO will trigger the bone marrows to produce more red cells. Okay, so if I draw it out for you, it's like this. Right? Athletes sometimes do this. Okay, they move to a higher altitude to train, where there's a thinner air, less oxygen in the atmosphere. So your body has lower oxygen, right? Okay, that will trigger the kidneys to release EPO. EPO will trigger your stem cells to make more red blood cell, and then more red blood cell means you will now be able to carry more oxygen. Okay, so that's what EPO is. Now you can synthetically engineer to manufacture EPO, okay, using the biotechnology that you learned about last semester. And EPO is actually used for cancer patients who are anemic because of chemotherapy and radiation therapy. Okay, because the bone marrow is a site of active cell division, and a lot of these chemotherapy and radiation therapy, they're targeting rapidly dividing cells like the cancer cells, right? So it's common for cancer patients receiving these treatments to experience anemia. Okay, and so giving them a little bit of EPO would help counteract that effect. Same thing for a patient who are undergoing dialysis. Dialysis involves taking the blood out of the patients, right, whose kidneys are experiencing some form of failure, and then cleaning the blood and then returning it to the patient. So the red cells, some of them actually ruptures and die as they pass through the tubings and all that, and it's not uncommon again for these people to experience anemia. So giving them a little bit of EPO would help. Okay, so just to reiterate, you make your own EPO when you have low oxygen in the body, right, to boost RBC production. But you can also make it in a lab and give it to people who need it, such as cancer patient or people using undergoing dialysis. Red cells don't live forever. They have a lifespan of about 120 days. So if you go donate blood, they tell you, you gotta rest about 3 or 4 months before coming back for another donation. You can donate just plasma, and if you do that, you can go more often. Okay, but if you donate whole blood, you gotta wait, 3 4 months before going back. And that's because you gotta wait for new blood cells to be made. Okay, so roughly, they live for about 120 days. When they're too old, the red cells will get trapped in the liver as well as the spleen. I already told you where your liver is. Your spleen is on the opposite side. Let me just go back here just to briefly show you. You don't have to go back. You can just watch. Okay. So liver is over here. The spleen is over here. Okay, you have one spleen. Just kind of tucked behind the stomach on the left side. Kind of smaller organ, much smaller than the liver for sure. So the liver and the spleen is where you will break down these old red cells. There are some macrophage, some white cells there to eat them up. And when you break down the red cells, you will release the hemoglobin. Now, the hemoglobin is precious resource. We don't want to just, completely get rid of it. We want to recover the iron so we can use the iron later on. And the protein portion is going to get metabolized, and eventually it becomes something called the bilirubin. Okay. So iron portion gets returned to bone marrow, so we can use it again to make another hemoglobin. The remaining protein portions gets broken down into something called bilirubin. Now, bilirubin has a yellowish color to it. Okay, yellowish color, and that's the thing that makes our urine and feces the color that they are. Okay, now sometimes you have a lot of red cells dying in the body for whatever reason. We'll get into the reasons later, and your body is not able to get rid of all these bilirubin fast enough. Okay, so a lot of red cells die, meaning you have a lot of hemoglobin that you need to metabolize, and when you do metabolize them, you end up having a lot of bilirubin. Okay, and if you have a lot of bilirubin, sometimes your body cannot get rid of it quick enough, and they start to build up in your skin. Okay, and the body kind of have this yellowish hue to it. So what do we call it? What's the name of the condition when someone has a yellowish skin buildup in their skin? Anybody know what that's called? Like, it's common in babies, right? When they're first born.
Jaundice?
Jaundice, that's right. Okay, so Jaundice. That's when again, there is a buildup of bilirubin in your skin, right? Even the eyes can turn a little bit yellow, right? And so we're going to come back to this later on in the lecture. Maybe not today, probably next class, okay? And discuss more about bilirubin and jaundice. Okay, so this, again, it's something that is common in newborns, usually due to mismatch of blood between the mother and the child. More on that later. So next we have the white blood cells. White blood cells are also produced in the bone marrow. And the fancy name for white blood cells is called the leukocyte. Leuko is white. Okay, leukocyte. Unlike the red cells, which are confined within the blood vessels, the white cells can actually move out of the blood vessels. Okay, you will find white cells in some tissue fluid. That's the fluid between the cells. You will find them living in certain organs, particularly organs that are very prone to getting infections. Okay, things like your lungs. Your lungs is constantly being exposed to the external environment, right? Every time you breathe things in, you might breathe in some pathogens, right? So in the lungs, there are some white cells that permanently lives there. Their job is to do tissue maintenance, to make sure that your tissues are not damaged, and if there is any kind of damage caused by pathogens, they will start to fight them off and perhaps initiate the repairing process as well. But they also do a little bit of housekeeping tasks, right? In the last slide, we saw that in the liver, we have some type of white cells that would destroy old red cells. Okay, so they are also responsible for destroying dead cells as well as dying body cells. And some cells are, they divide too many times already, and they're no longer carrying out their function efficiently. So you got to kill them off. And that is part of the job of the white cells as well. Okay, so here in this picture, you can see there's the white cell. They're not actually white, by the way. Like not literally anyways. They are all like see-through, right? But compared to the red cells, they are kind of colorless. That's why they're called white blood cells. But when you stain them and look at them, they usually have these purple colors. Okay, so the white cells can squeeze through the pores in the walls of the capillaries, and then they would go out and do their job. And what exactly is that job? We will learn more about it in the slides to come. Now, there are two groups of leukocytes, two groups of white cells. Okay, the red ones here, the circle, the one that are circled in red, those are called the granulocytes, and then the other ones we call them agranulocytes. Okay, like you don't have to be able to tell which one is which. Okay, your job is to know that there are two groups: the granulocytes and the agranulocytes. So let's take a look, take a note on this, and explain the difference between the two. Okay, so we have the leukocytes at the top. Leukocytes, our WBC, white blood cells. So the leukocytes are separated into two categories. We have the granulocytes and we have our agranulocytes. It's the exact same word, okay? With the extra A at the top, at the front. Agranulocytes. Okay. So what's the difference, right? Granules are kind of like lysosome. You might remember learning that last semester, right? These are sacs that contain enzymes and chemicals. Okay, it's kind of like a lysosome. So what is the difference between the granulocytes and the agranulocytes? They actually both have granules, but the granulocytes are the one that has visible granules under the microscope. In other words, you can see them when you stain them and look at it under the microscope. The agranulocytes, the granules are not visible under microscope, even though they exist. Okay, so it's just like a visual distinction. If you can see them easily, when you stain them, we call them granulocytes. When you cannot see them, we call them agranulocytes. Okay, so it's a visual distinction for people who look at these slides all the time, right? You want to have a quick way of classifying them. That's all. So let's take a look at some examples of each. What color did I use in the slide? Okay, red is the granulocytes. Okay, so granulocytes. Right. One example of granulocytes is neutrophil. Neutrophil is the most abundant WBC. Right. 70% of all your white cells are neutrophil. It sounds like a lot, right? But remember, all the white cells only make up 1% of all your blood. So of that 1%, 70% of it would be neutrophil. So what do they do? A neutrophil is a type of phagocyte. I know it's a lot of new terms here, okay? But try to stay with me, and if you don't understand, let me know. A phagocyte is a cell that can do phagocytosis. You remember phagocytosis from last semester, right? Okay, like you have a cell here. Let's say there's a bacterium. And then if this is a phagocyte, it will basically like eat it up, right? Like that. That is phagocytosis. So any cells that can do this is called a phagocyte. Okay, so I'm just going to put it down here. Phagocytosis, like Pac-Man, right? It eats up the bacteria and then it's inside now. So neutrophil is a type of phagocyte. There are other phagocytes that we'll learn as well. It's okay to classify a cell with more than one label. Okay, so a neutrophil is both a granulocyte and it is also a phagocyte. Right? Visually, it's a granulocyte because you can see their granule. Functionally, it is a phagocyte because it goes around and eat things up. Okay. So that's what it is. It essentially goes around and remove foreign substances. If it's not supposed to be in the body, it will eat it up. That's neutrophils. And we have another one. Not everything that doesn't belong to the body can be eaten up. Some of them are really big, right? Someone mentioned about tapeworm earlier. There's no way a cell can eat up a tapeworm, right? It's like so much bigger than itself. And so we need to have another type of granulocytes, which we call eosinophil. Eosinophil. Okay, so rather than eating up the pathogen, rather than doing phagocytosis, the eosinophil, they will secrete chemicals from the granules to attack the parasites. Okay, it's too big, can't eat them up. I'm just like summarizing it in very simple terms, right? Of course, they have other functions as well. So if you read in the online textbook or whatever that they have other functions, great. But you only have to know the one I'm writing for you. Okay, I'm just gonna do one more. This is basophil, and the function of basophil is to plays a role in inflammation. And we're going to learn more about inflammation later, but basically, think about last time you cut your finger accidentally, it's gonna hurt, is gonna be red, swollen, right? It feels a little bit warm. All those are characteristics of inflammation. And in part, that's caused by the basophil. All right, so there you have it, 3 types of granulocytes. Now, on the other hand, we have the agranulocytes. They have granules, but we just can't see them clearly. And so one example is what we call monocytes. Okay, so these monocytes, they patrol the tissue and the body, just to see what's supposed to be there and what's not. They're kind of like the security guard of a building. Okay, they move around, make sure everything is okay. And if they encounter something that does not belong to the body, they can transform. They can differentiate. Differentiate means they change and specializes, differentiate into macrophage. Okay, so this is another type of phagocyte. Okay, it's worth mentioning that. I'm going to put a star here for you. Put a star here. Some tissue have resident macrophage in them. Okay, because they are vulnerable to infection, like the lungs I told you, right? So you don't have to wait until there's something wrong to transform into the macrophage. Okay, they're always going to be macrophage in them. They live there. That's why we call them resident macrophage. Okay, all the time. And you will have some in the liver, right? Always need to be there to catch the all red cells, for example. Okay, I'm just going to give you two more. The next one is called a T cell. There are different types of T cells that we'll learn in lecture 3. But for the most part, T cells, they help coordinate immune response. Okay, it'll tell things what to do. Okay, you go attack, you change to macrophage. Okay, and then you go make antibodies. That's the function of T cells. It'll do a little bit of killing itself as well, but that's what it does for the most part. And we have the B cells. B cells is like our ultimate weapon to defend the body against pathogen. They make antibodies. These are the Y-shaped proteins that are used to attack pathogens. Okay, so there you have it. 3 granulocytes and 3 agranulocytes. All right. I hope that makes sense to you. Of course, I'll post this later on when we finish the whole lecture. But let's move on for now. And there's only one last thing to talk about, which is the platelets. Okay, platelets also has a fancy name. They all have a fancy name. This one is called the thrombocytes. Thrombocytes. Erythrocytes for red, leukocytes for white, thrombocytes for platelets. Now, platelets are actually not really a cell. They are more like cell fragments. They don't have a nucleus. They don't have a lot of the organelles. They don't really function on their own. Okay, they're just like fragments that are broken off from a bigger cell, and that's the big cell right here in the middle. Right, it is called the mega. Mega means really big, right? Mega karyocyte. Okay, and all these little things that are breaking off from it, those are your platelets. You make a lot of platelets every day. Okay, an astounding rate of 200 billion platelets per day. And a lot of them actually would just end up being broken down within 10 days or so if you don't use them. Okay, and for all intents and purposes, we don't really want to use them because the only time we need the platelets is if we are injured, right? If we have a cut or something like that, right? So the function of platelet is hemostasis. Don't confuse this with homeostasis. Homeostasis is internal balance, right? Okay. Hemostasis is basically to stop the bleeding. Okay. So that is platelets. Where do we get platelets from? How do we, what part of the body makes platelet? Do you remember? Well, platelet is the cellular component of blood, right? And all cellular component of blood came from the bone marrow. Right? So I'm just gonna go back here to show you. Right? You have your bone marrow stem cells, and they could become a red cell, they could become platelet, and they could become any of these white cells that we talk about, right? Basophil, eosinophil, neutrophil, so on and so forth. Okay? Let's have a quick note on coagulation. Oops, drop my pen there. So, when you damage your tissue, coagulation. Coagulation also means stopping the bleeding, right? So hemostasis, coagulation, they're kind of used interchangeably for us. And so in your plasma, you have fibrinogen and prothrombin. We saw these earlier in one of the earlier slides. And so these are plasma protein necessary for forming a blood clot. Right? Now, try to learn some of these suffix and prefix. Ogen. Ogen means it is inactive. Okay. Pro also means it is inactive. So unless you have a cut right now, or if you have some kind of bleeding going on, your fibrinogen will stay in the inactive form. Your prothrombin will stay in the inactive form. And we only want to activate them if we need to stop some kind of bleeding. Now, this is again an oversimplification, but it will be enough for our purpose. So let's say there is tissue damage. Tissue damage. Okay, so tissue damage is accompanied with like cellular damage and stuff like that, right? So when a cell is damaged, it will release something called thromboplastin. Thromboplastin. Okay, thromboplastin. So we don't have thromboplastin unless tissue is being damaged. Then the prothrombin, which again is inactive, will get turned on, will get switched on in the presence of thromboplastin, plus a little bit of calcium. That will turn it on. And when you switch it on, it's going to be called thrombin now. Right? This is the active one. Okay. And then the fibrinogen, which is again inactive, will get converted into fibrin. In the presence of thrombin. Right? And you also need a bit of calcium for this. And then the fibrin will form these long strands of fibers. Those are the fibrin. And it will help trap, red cells. It will help trap platelets. And eventually it will form the blood clot that will seal the wound and stop the bleeding. Okay, so there is a series of events, a cascade of events that ultimately lead to the creation of the fibrin. And that's the scab, right? Sometimes you have a scab and you pick on it and you bleed all over again, right? The scab is essentially a mixture of fibrin, red cells and platelets. And all begins with the tissue damage releasing the thromboplastin. Okay, does that make sense? So there might be a question on the test that lists all these steps for you, and then you have to rearrange it and tell me what happens, right, in the blood clot. Now, there's something called a PTT, okay, a thromboplastin test. And a PTT test basically tests for how long does it take to form blood clot in a person. Okay. And that information is useful for someone who may have to go in and have surgery. Okay. We don't want the person to take too long for the blood clot to form, right? Okay, so examples of why a person might have long PTT is maybe their prothrombin or fibrinogen level is low. Okay. Maybe the person is on blood thinner, right? Like warfarin, for example, right? Aspirin, we talked about aspirin is a blood thinner last time, right? Yeah. We don't want PTT to be too short either. If someone's blood clots very easily, then that person is at risk of getting stroke, heart attack, right? Because we don't want blood clot to be forming in one of these capillaries, for example. Are we okay? Any question at all? Please let me know. I know I've been talking for an hour. I didn't really ask you guys any poll questions. But are we together? Okay, well then. This is a summary of everything we've talked about. Obviously, well, maybe not obviously, but like you don't have to memorize the numbers. Okay, you don't have to know, you know, 46 million to 5.4 million is red cells or something like that. Okay, you just have to read through the main function and the main component and see if you understand them. Okay. All right, let's do some polls. Try to do it without looking at your notes to see how much you remember. Which of the following is an example of granulocyte? All right, anyone else? I have 11 attempts out of 20 people who are here. Last call before I end it. Okay, so most of you got it right. It's the basophil. Basophil is responsible for playing a role in inflammatory response, inflammation. And the rest of them would be agranulocytes. Let's try another one. Okay, thank you for participating. That's correct. It is the parasitic infection. Eosinophil will secrete chemicals to attack things that are too big to be engulfed. Another one here. Yeah, everybody got it. It is found in the RBC. Very good. That's it. Yeah. Let's take a 5-minute break and we will resume at 9:41. When we return, we will learn about the genetics of blood. Okay. See you in a bit.
Sir,
Yeah.
can you leave the screen on the notes?
This one? This note?
Yes, thank
Okay. All right. See you guys in a bit. Hello. Let's keep going. What's going on next? Let's see. Blood genetics. Okay, so let's talk about what exactly is blood type. Does anybody know their blood type? And you want to share, perhaps? Anybody know their blood type? You know what? Why don't we do a quick poll? Okay, just to see if you know. And like A, B, O, don't know. Okay, like A, B, C, D, E. Let's do a poll here. Let me know. You know your blood type. Okay. Yeah, that's pretty interesting. Okay. Well, I have the usual participant. So of the 10 people who participated, this is the distribution. Okay. So some people don't know, and we don't have any A's here. It appears. But what is blood type? Okay, and a lot of people don't just tell you that you're A or B, right? You actually would say I'm like A positive or A negative. Some of you might not know whether you're positive or negative, but you know you're A, B, or ABO. So what exactly are blood types? Blood types refers to the inherited antigens present on the surface of red blood cell. First of all, it is inherited. That means you don't get to choose it. Okay, you're born with your blood type. And antigens, okay, you should write this down. Antigens are basically molecules found on a cell, any cell. Okay, it doesn't have to be red blood cells per se. But when we talk about blood type specifically, then we are talking about the molecules on the red cell. Okay, so these kind of molecules can be proteins, it could be carbohydrates, could be glycoproteins, which are like sugary proteins. They are basically proteins with sugar molecules on them, or glycolipids. Okay, sugary protein or sugary lipids. Okay, lipids with sugar molecules on them. Okay, like short saccharides on them. And depending on whether you have certain proteins or not have them, you would be a particular blood type. And so when you say you're type A blood, then on the surface of your red cells, you have these A proteins. They're actually glycoproteins. Okay, A glycoproteins. These are inherited. That means they are genetically determined. So you would have a gene that makes the glycoprotein A. Okay. In the case where the person is AB, then they would have both protein A and protein B on the surface. Now, ABO is only one of the many blood groups that exist in the human population. When you say you're positive or negative, you are actually talking about an other blood group called RH. Okay. But in addition to ABO and RH, there are over 40 blood groups that exist, right? If I take you to the Wikipedia page here, they're all listed here, right? So there's the ABO blood group that I just talked about. There's the RH blood group, but there are many more. MNS, right? Cal, Duffy, so on and so forth. And the list goes on and on and on and on. Okay. Now, in a real life situation, well, I guess I don't know if I should call it real life, but like in a situation where a person needs to have a planned blood transfusion, you will try to match all these blood groups, right, as much as possible. All right. It's only in an emergency situation where you got to give blood to another person, otherwise they're gonna bleed out and die. Then you would only look at the ABO and the RH. Okay. But in a planned transfusion, you would have to match the blood groups completely, okay, to avoid incompatibility. But for the most part, much of the remaining blood groups are quite it's not as diverse, right, in the human population. So they're very similar, especially if you're the same ethnic group. It's only the ABO and the RH that are more diverse, and that's why those are the main ones that get matched in an emergency situation. Okay, so we'll talk more about that later. This is according to the Canadian blood surface. Most Canadians are type O positive, okay, followed by A positive. And B minus is quite uncommon. Okay? I am AB positive. I'm AB positive. All right. I'm not sure why you would want to know that, but that's what I am. So let's take a closer look at the ABO system. Okay, then we will learn RH. Probably not maybe today. I don't know. We'll see how much time we have. Okay, so we'll do the ABO system and then RH for the lecture. The rest of them we can just kind of ignore for now. So ABO blood type is based on the presence or absence of the two antigens. Okay, so do you have it or do you not have it? If you have A antigen, the protein A, then you will be type A. If you have B antigen, then you will be type B. If you have both of them, then you will be type AB. If you have neither of them, then you will be type O. Okay, so there are 3 alleles that is responsible for determining our blood type. Alleles are different versions of a gene, right? We learned that last semester. So there's the IA allele, IB, and the little I. So the IA allele, the gene, allows the person to make, we'll just call it protein A for simplicity, okay? And then this one allows the person to make protein B. Okay? And then little I makes nothing. Makes nothing. IA and IB are dominant. A dominant over little I. Okay. But IA and IB are co-dominant with each other. Okay, in case you forgot, dominant means it will cover it up. Co-dominant means both of them will be expressed. Okay. The best way to think of this in terms of an analogy is, think of cookie cutters. Right? Let's say you have 2 types of cookie cutters. You have moon cookie cutters and star cookie cutters. If I only give you moon cookie cutters, you will only be making moon cookies, right? It doesn't matter if I give you 1 of the moon cookie cutters or 2 of them. If you have moon cookie cutters, you can only make moon ones. If I give you both moon and star cookie cutters, then you'll be making both types of cookies, right? And that's what IA and IB means. If you have both of the alleles, you will be turning out both protein A and protein B on the surface of the red cell. Okay, so it depends on which one you have or if you have both of them. And little I would be equivalent to not having any cookie cutters. So you won't be making any cookies at all. Okay. Any question about these alleles before we move on? Because that's all we'll be talking about for the next half an hour, 45 minutes. So if you need clarification, now is the time to ask before it gets too confusing. Okay. Blood type notes. All right, let's do this. we actually will use the study guides for our notes. Okay, so we're going to go to this chart right here. It's on the second page, question number 6. If you don't have the study guide readily available, you can easily make this chart yourself. Okay, so genotype. Genotype is the genetic makeup of the individual, right? So if someone is type A blood, what kind of allele would cause the person to have type A blood? Well, they could be IAIA. Oops, IAIA. Now, even though there are 3 alleles that exist in the population, because we are diploid, we can only have 2 alleles at a time, right? So IAIA or IA little I. So again, right? Think of the cookie cutter analogy. It doesn't matter if you have 2 alleles or 1 alleles. As long as you have one of the IA alleles, you will be making protein A. That would cause you to be type A blood. Okay, this doesn't necessarily mean you can make more. Okay, like it just means it's not about more or less anyways. It's about whether you have it or not. Okay, so what kind of antigens will you have on the red cell? Okay, so we're going to draw our red cell here. Okay, and because we have the IA allele, we will have type A protein on our surface. Okay. Now, just a quick note about the antibodies. Okay. We'll learn more about it next week, but for now, just remember, you will. We'll say you will. Let's say you can. Sorry, you can make antibodies that target foreign antigen. Okay. Now, in the past, students get very confused between antigens and antibodies. So I'm just gonna try to explain that one more time. Antigens are things that are found on a cell. Okay, don't think of them in terms of good or bad. They are just things that are found on a cell. Okay, so all your cells have antigens. All my cells have antigens. Bacterial cells have antigens, right? Okay, viruses, they have antigens. Okay, they are just things that are found on a cell, right? But your antigens will be different from my antigens. Okay, and in fact, your antigens will be different from the antigens of everyone else in the world, unless you have an identical twin. Okay, and so anytime your body encounters a foreign antigen, okay, something that is from the outside of the body, right? Your immune system will make antibodies to attack them, okay, that will target them and attack them, right? So if you are a type A blood person, that means you do not have type B antigen on the red cells. You don't have protein B, right? And so your body will make anti-B antibodies. Does that make sense? Anti-B antibodies. Let me write it smaller. Anti-B antibodies. Okay, so they are Y-shaped proteins, and I'm going to put the letter B at the end to show you that they recognize and attack B. Does that make sense? Okay. Like, you know, if you've gotten chicken pox and you've recovered, now you have anti-chicken pox antibodies, right? You make antibodies that would target anything that does not belong to the body. That's what foreign antigen means, right? It's like from outside of the body. Does that make sense? Okay, we will see if that makes sense in a bit. Type B blood. Okay, what are the alleles for type B blood? Help me out here. Anyone else? Anyone who wants to talk? What are the genotype for type B blood?
IA, IB, and IB and just regular I.
That's right. You'd be absolutely correct, right? Homozygous IB or heterozygous IB little I. All right, and this time, if we draw the red cell, there would be B antigens on the surface. Now, what kind of antibodies will we be making in this case?
Anti-A.
Anti-A, that's correct, Shelley. This would be anti-A antibody because your body will make antibodies that targets things that you don't have. In fact, you do not. You do not want to make antibodies that would recognize your own antigens, right? Because then you would end up attacking yourself. And that is a bad thing to do. Okay. Type O blood. Type O blood is when you do not have A and do not have B. And the only way for that to happen is little I, little I. So when I draw our red cells, it would just be that with nothing on the surface. Now, please pay attention for a second, okay? When I say there is nothing on the surface, I mean no A and no B, right? I'm just talking about the ABO blood group, right? It does not literally mean nothing on the surface. Because remember, there are like 40 different blood groups, right? So even though you don't have any A or B, you might have an RH on it, right? And that's talking about a different blood group, right? So we're just talking about within this ABO blood group here. So if someone is a type O, please think about it. What kind of antibodies will the person make? What kind of antibodies will we have in the plasma of that person?
Both anti-A and anti-B.
That's right, right? Because we don't have A, we don't have B, both would consider it as foreign. And you would have anti-A as well as anti-B antibodies. Okay, I don't have space to write the word antibody, so I'm just going to put anti-A and anti-B. All right. Next, we have AB. So IA, IB, that's the only possible genetic makeup for this. And the person will have both A and B on the surface of the red cells. And in this case, it will not be making any antibodies against A or B, because then you would just end up attacking your cells, right? And I told you we do not want that to happen. Okay, so for someone who's AB blood, then no antibodies for them. Do we have any questions about the genetic makeup and the relationship between antigens and antibodies? You need to understand this before we talk about compatibility with blood donation. Okay, let's talk about blood donation, and I'm gonna need a new nook for that. Okay, so this is for RBC blood transfusion. Okay. Just give me a second. I'm gonna check my PowerPoint and make sure that I'm not going ahead of myself. Okay, we're good. Oops. Okay, so we do need this note. RBC blood transfusion. I told you earlier today, when you do transfusion, you have to separate the red cell from the plasma, right? So we are focusing on the red cells. And in fact, that's all we're going to talk about, right? In the past, I taught both red blood cell and plasma transfusion rule, but it confused students like tremendously. So I'm just going to talk about RBC transfusion. All the questions on the test, if they just say blood transfusion, you can assume I'm talking about RBC blood transfusion, okay? So there are 3 questions you must ask yourself. Question number 1, what type of antigen? Okay, you know what? I'm just gonna keep it short. Okay, the first question is antigens on donor's RBC. Right. Second question, antibodies in recipient. The recipient is the person who's receiving the blood. Plasma. And last one is, will the antibody attack the antigen? Okay, the RBC. Those are the three questions that we have to ask ourselves. Okay, so let's say we take AB blood and we give it to A. Okay. Do you think this is safe? Safe or not safe? Okay. Don't think too much, just like, you know, gut feeling. Okay. We will talk about how to use these questions to answer it in a second, but you know, just off the top of your mind, do you think AB blood can be given to A? Okay, anyone else? I'm curious, how come you guys not doing the polls? Like, is it because you guys cannot see the poll, or is there some other reason? Because this is very important for me as feedback to see, you know, whether you guys understand or not.
Sir, did you mean AB positive or just
Oh
AB?
no, we're not talking about positive or negative. We're just talking about ABO for now. Okay. Anyone else? Last call. All right. This is the perfect result that I want for a question like this, right? 50-50, right? So that's before I teach you, right? After I teach you how to do it properly, I expect all of you to get the right answer, of course. But now, you know, is it safe? Is it not safe, right? Half of you think it's safe, half of you think it's not safe. So excellent. Let's find out how to do this properly, okay? And again, thank you for participating. So we're going to take AB blood and we're going to give it to a person who's A. Okay, this is a donor and this is the recipient. By the way, I want to warn you, okay? Some of you might have heard this phrase before. Universal donor, right? Universal recipient. You probably heard something like that before, right? Okay. I will tell you why I don't like that term and why it is confusing. And you should not be thinking about those things when you try to answer these transfusion questions, okay? Because it will almost always mess you up, okay? So ignore those universal donor and recipient things that you might have heard in your life and just try to do it like the way I'm teaching you right now. Okay, so number one, antigens on the donor's RBC. So I'm going to draw it out, okay? So this is a person who's AB blood, which means on the surface there will be A and there will be B. That should make sense to you, right? That's the most straightforward thing. So that's number one. Number two, we got to look at the antibodies in the recipient's plasma. The recipient's plasma. Okay, so the recipient is type A blood. What kind of antibodies, if any, would be found in the plasma of someone who's type A blood? If you don't remember, you can look at the chart, but try to think about it. Right? What kind of antibodies does a type A person produce?
B.
B is correct, right? Because you make antibodies that would target something that you don't have. So if you go ahead with this transfusion, if you take the red cells from the donor and give it to the recipient, all these antibodies will recognize the B, right? And it will attack it. It will attack it. So this is not safe. Not safe. Does that make sense? Those of you who chose safe, have I convinced you why this is not safe? I certainly hope so. Let's try another one. Okay, let's try another one. If we take type B blood and give it to type O. You know what? Let's do another one. Let's take a type B blood and give it to AB. Okay. Is this safe or is this not safe? Let's try to do the poll again. A for safe, B for not safe. Okay, so either I'm not teaching properly or there's some kind of confusion going on, because this is also not very good results. Okay, let's do it one more time. Let's do it together. All right, and please ask questions. I don't see, the thing about online learning is I cannot see your faces. So I don't know like what kind of confusion you might have, or like if you're confused at all. So please communicate, talk to me and let me know what you're thinking so I can help you understand better. Okay, first question. Antigens on the donor's RBC. So donor, what letter should I put on the surface of the cell, huh? What should I put here? B, right? Do you understand? Because this is the donor. That's the donor. This is the recipient. Okay, the donor has it's type B blood, so I will put the letter B on it. Okay. What about the recipient? Okay. What kind of antibodies, if any, does the recipient have in the plasma? Remember, the recipient, the person who's receiving the blood, is AB blood. What kind of antibodies will we have?
They don't have any.
They don't have any. That's right. No antibodies, nothing against A or B. So that's okay. That's safe, right? That's safe because if you don't have any antibodies, you can give them any red cells and there will be no reaction. With no antibodies, there will be no attacking at all, right? So that's why this is going to be safe. Okay, I'm going to pause here, right? I'm going to take some questions. And I know there are some questions. There is no stupid question, okay? If you are thinking about a question, I guarantee you like 3 or 4 other people are thinking about the exact same question. So please ask so that I can help you. Yeah, go ahead.
I chose not safe, but I realized my mistake. I was looking at the antigens instead of the antibodies.
Okay, yeah, very good. So it's good that you caught your own mistake and hopefully, it'll be okay moving on, right? But that's a common misconception, right? Confusing the two. Anyone else have any questions, comments, remarks? You will have a lot of these questions on the test, so I just want to make sure you are okay with it. Okay, I'm just going to do one more and hopefully we'll get 100% correct rate this time. We're going to go ahead and take B and we're going to give it to O. Okay, is it A safe or B not safe? Okay, it is improving, so that's a start. Let's go through it together. This is the donor, and this is the recipient. Okay, so again, we have to ask ourselves the 3 questions. What kind What kind of antigens do we have on the surface? Well, if the person is a type B blood person, we will have B on the surface. Okay, for the recipient, what kind of antibodies do they have? This is a type O person, so there will be type A, anti-A antibodies and anti-B antibodies. So the anti-B antibodies will attack. Basically, if the antibody has the same alphabet as the antigen, then there will be incompatibility, right? They will start to attack each other. So for that reason, this is not safe. All right. So you probably want a cheat sheet, right? When you write the test, you probably want to have a sticky tag on your computer screen with this information on. Okay. Again, it's not cheating. Okay, I'm giving you permission to do that. All right. So O is at the bottom, and you can give O to A, you can give O to AB, and you can give O to B. And then A can be given to AB, and B can be given to AB, like that. Okay, so that's the transfusion chart. Of course, O can be given to other O's as well, right? B can be given to other B's, and so on and so forth. Okay, so using this chart, you should be able to tell what's a compatible transfusion and what's not. Okay. So what about this one? AB to B, safe or not safe? Okay. What do you think? A for safe, B for not safe. I mean, it's much better, but still not 100%. I'm not sure what the confusion is. A, B to B. If you look at the cheat sheet, AB cannot actually give to anybody. The only other people AB can donate to is other ABs, right? There's no arrow pointing anywhere else, right? So AB does not point to B. Therefore, this is not safe. Okay. In terms of the explanation, and you should be mindful of the explanation, AB means they have both A and B on them. For someone who's type B blood, they would have anti-A antibodies. And if you go ahead with the transfusion, the antibody will attack. And that's why it is not safe. Okay, so I think we're gonna just go back to our chart right here. I'm gonna give you a moment to fill this last column in, and then we'll go back and do the PowerPoint slides. Okay, just gonna give you 1 minute to fill this in. You can either think about it using reasons and logic, or you can use the cheat sheet to help you. So remember, you can always donate to people of your same type. Okay, so if we just look at the ABO system, A people will be able to give to A. And you follow the arrow and see where it leads to. You can also donate to AB. Okay, so the answer for this chart is as follows. So O can be donated to everyone else. And that's why sometimes they're called a universal donor. That's the reason, okay? But as I will show you later, that is actually not entirely accurate. Some of you probably know why already. Any question before we move on? Seriously, don't be shy. Already, let's move on. Let's go back to okay, I got a private message. Only O can give to O. Yes, that's correct. Only O can give to O. All right, we're gonna do some genetics problem here. A man with AB blood is married to a woman with AB blood. What blood types will their children be, and in what proportion? So you already know there are 4 blood types, but only 2 of them has one single genotype, okay, which is the AB and the O. If I tell you I'm type A blood, you don't know if I'm homozygous, IAIA, or heterozygous, IA little I, right? But if I tell you I'm type O, then you immediately know I am little I. Similarly, if the person is type AB blood, then the genotype is IAIB. So in this case, the man is IAIB. And the woman as well, okay, IAIB. So they have matching blood type. How sweet. What's going to happen is they're going to have some kids, and we want to know what proportion will the kid's blood type be. So this is exactly same as the genetic unit last semester. The man, half of the gametes will have IA allele, and the other will be IB. Okay, because in meiosis the chromosome separates, right? And the woman will be same thing, IA and IB. It doesn't matter where you put the man and where you put the woman. Okay, I'm gonna put the man here. I'm gonna put the woman here. Those can be interchanged. It won't change the results. So let's take a look. If this sperm fertilizes this egg, then we are gonna have IAIA. If this combines with this, we will have IAIB. Right. This one, also IAIB, and then finally IBIB, right? It's just filling in the Punnett square like you did last semester. And so in terms of proportion, there is a 25% chance they will have a IAIA, which is of course type A. There's a 50% chance that it will be IAIB, which is type AB blood. And then finally, there's a 25% chance that it will be IBIB, which will be type B blood. Okay, so that's pretty straightforward. Any questions? Right, let's move on. One of the fears my wife had when we, had our first baby was that we're gonna take home the wrong baby, okay? Because there's all these stories you read that they sometimes take the baby away, and then they brought back the wrong one, and then you end up raising your wrong kid, and then you're realizing it 40 years later or something like that, right? Okay, but actually, you know what? She told me, your job is to stay with the baby at all times. As soon as the baby comes out, you just stay with it. Wherever the baby goes, you go. Right? So that was her mission for me, right? And I said, but what about you? Wouldn't you need me to help you? Or are you going to be like bleeding and in pain and all that? She's like, don't worry about me. You go with the baby. Wherever the baby goes, you goes, right? And then we found out, that was that concern was irrelevant, because they actually never take your baby away, not in Canada anyways, okay? her sister, which was gave birth in Hong Kong, they actually do take the baby away to examine and do tests on it and then bring it back. But not in Canada. You stay with your baby at all times. And I'm pretty sure we brought back the right baby because my daughter, unfortunately for her, looks exactly like me. So there's no problem in that. But suppose there is a mix-up, right? And in the hospital, and you know, can we use blood types to determine which baby belongs to which parents? And that's what this question is. Right, so we have two babies here. Baby 1 is type O and baby 2 is type A. And we have two potential parents here. The James, the blood types are these, and then the Marshalls have these blood type. So can you use blood type alone in this particular example to figure out which baby belongs to which parent? Okay, so I'm going to give you a moment to think about that. And I'm going to do another poll for you. Okay, I'm going to be asking for baby 1. Okay, baby 1. Does baby 1 belong to the James or the Marshalls? Tell me what you think. Okay, interesting. We have a lot of people saying that the baby 1 belongs to the the other one is climbing up as well. Okay, so about 50-50 again. All right, so let's take a look at how to answer this question. All right. So baby 1 is type O. There's only one way to have a blood type of O, which would be little I and little I. All right, so let's take a look at the James, right? Mr. James here is type AB. So type AB is IAIB. So if you look at that, immediately I could reject the first baby as their kid, because Mr. James the father does not have a little I to pass on, and you need two little I's to make a type O baby, right? So it's impossible for someone with type AB blood to have a type O child. They do not have a small I to pass on, right? And so as a result of this, we can reject baby 1 as their kid. And so baby 1 belongs to the Marshalls and baby 2 belongs to the James. Now you might wonder, how come two type O B person can have a type O baby? Shouldn't they be, shouldn't they be like IB? Well, not necessarily, right? If they are IB little I, then there's a 25% chance that they will have a type O baby. That's the reason. Do you understand? Yeah, O has to come from two little I. So each parent must donate a little I in order to make a type O baby. Are we together on this? By the way, like in real life, nobody's gonna, use the blood type to figure out whether the parents, belongs to the baby or whatever, right? That's not accurate. This is just for like, academic purposes. Okay, we to make sure to find out what baby belongs to what parents. If there is a mix-up, we do DNA testing, right? Okay, that's like 99.99% accurate. All right, let's up this a notch, make it more complicated. There's a question right here in the study guide. Wait, sorry, give me a second. I haven't used this note for some time, so I'm a little bit confused sometimes as to what question goes where. This is B. Okay, this is a different one. Right. Okay, so let's do this together. Now, oh, I have a question in the chat. Type A and type B parents will have, okay, you know what? That's actually the next thing I'm gonna talk about before we attempt something like this. Oh, we cannot do this yet. I'm sorry. I still have to talk about the plus and the minus. I'm all over the place. Please forgive me. Okay, we'll come back to this later. We haven't learned RH yet, so we cannot answer that. And Emma, for your question, just hold that thought, okay? We'll come back to it later on, okay? Because there are still more things you have to learn before you can answer that. Okay, moving on. Genetics problem number 3. Okay, this is a pedigree of the Shrek's family. You might have seen this cartoon before. So I put down everybody's genotype and blood type rather. And I want you to figure out the blood type and a genotype of Fiona. Okay. In case you need a refresher on pedigree, square is male, circle is female, horizontal line means to get married, and if you have a line coming down, that means they have kids. Okay. So Shrek and Fiona had 3 kids. Okay, 2 boys and 1 girl. Please figure out everybody's genotype, especially Fiona's. Okay, so this I'm gonna give you like maybe 3 minutes to try, and we will take it up. When you do have the blood type, please type it in the chat box. All right, so I have a few people who have given some answers. We will take it up right now. I'm just gonna copy this and put it in OneNote, so I have more coloring options. Here. Okay, so to do something like this, we would start with people who are type A, B, and type O. Okay, those would be the ones that we know for certain. So that's IA, IB for the king. IA, IB at the king for the king. And Fargo here is going to be little I, little I. All right. And the next thing we're going to do is we're going to fill in half of what we know for everyone else. So queen is IA something. We don't know if she's IA or IA little I at this moment. Okay, we're going to find out soon. Shrek here is going to be IB something. Right? Fergus, IA something, and Felicia is IB something. Does everybody understand my approach? You fill in all the A, B's, and the O first, and then you fill in half of everyone else. Right? So that requires no thinking. You just do it, and then you analyze. All right. And there are a couple ways to approach this. I'm gonna use this particular approach. We're gonna start with Fargo here. He is little I. So that means one of the little I must have came from the father. Right? The other little I must have came from Fiona, the mother. Make sense? Because each parent has to contribute a little I, right? Where did Fiona get her little I? Well, certainly not from the king, right? Because the king can only pass on IA or IB. So the queen must be IA little I. Okay. All right, let's move on and do more analysis. Fergus. Fergus has an IA. Where did Fergus got his IA? Well, not from Shrek, right? Shrek doesn't have an IA. So the IA must have came from Fiona. So Fiona is IA, right? So anybody who said Fiona is IA little I, congratulations, you're correct. What about the other half? What did he get from Shrek? Did he get the IB? Did he get the small I? Well, since he type A blood, has to be the small I, right? Because otherwise, if he got the IB, then the blood type would be AB, right? For Felicia, IB came from the father, and then the other half has to be small I as well. Does that make sense? Okay, so you will have some chance to practice in the study guide, and we'll do some more poll questions later on with a pedigree. But that essentially is how you would approach a question like this. Okay. All right, fantastic. Moving on. Oh, we have some practice right now. Okay. Let's take a look at this, and I will launch the poll. Right. This one probably will take a little bit of time to work it out. No, a lot of people chose B as the answer. Let's see if you guys are correct. Right. Let's start with Ed here. Ed is type O, so Ed is little I, little I. Sue is also little I. Mary is IAIB. And then we're trying to find the genotype for John here. Okay. Well, if John and Harriet has a little I, then John must have at least one little I, right? And Harriet will be IA small I. So what about the other one? We don't have, well, I mean, we could write out the information for Tom and Jane as well, but we already have enough information. Right? If you look at Mary, the IA came from the mom, and the IB must have came from John, right? So that's the correct answer. Okay. Let's do one more. Same family, but now Mona. What is the blood type for Mona? Anyone else wants to choose something? You're gonna have to do questions like these on the test, by the way. So it's good to try it out now. Okay, we have a lot of people who said IA, IB for sure. Why is that? Well, this is puzzling. Okay, so previously we determined John is IB little I, right? So I'm gonna put IB little I here, and Harriet is IA little I. And so Ed is small I, which means Harriet got the small I from Ed, right? And the IA would came from Mona. But we don't know what the other half is. It's impossible to tell. This could be IA, right? It could be IA small I. It could also be IA IB, right? There are three possibilities here. And we don't know which one is the correct one. And so the correct answer for this case is need more information. Do you guys understand? For people who chose A, did you get confused? Like, did you thought John was like their son or something? Probably why, right? You're like, oh, the IB came from Mona. That's probably what you think, right? Okay. But John is not the part of family, right? Like, you don't marry your own siblings, right? Okay. So like Harriet is the only daughter. Okay. That's probably why you got confused. Okay, antigen antibody. We actually took the note already. I did it out of sequence. I should have waited until here, but that's okay. We took the note already on that. That's the chart with the antibodies and the red cells. And here is what we call a cross reaction. Okay, cross reaction. That's what happens when the antibody recognizes the antigen and why it is not safe. So here, instead of using the letters A and B, they use shapes to represent the surface antigen. So for someone who has type A blood, we have the circles here. Okay. And then for someone who has type B blood, for someone who has type A blood, they represent it with a little circle here. So in the first case here, they are taking type A blood and then giving it to another person who's also type A. Okay. And so the recipient, because they have type A blood, they will have anti-B antibodies. And you can see the anti-B antibodies, they have these little triangular groups. So the antibodies don't recognize the antigen, and as a result, there's no binding, there's no targeting, no reaction, and mixing the blood will be fine. Okay. In the second situation here, we have type A blood being given to someone who is type B. So the person with type B blood will have anti-A antibody. You can see the groove in the antibody is now compatible with the circle. So if you go ahead with the transfusion, the antibody will bind to the incoming cells, and it will cause agglutination. Okay? Agglutination. Agglutination means clumping together. Clumping together. And that will result in a cross reaction between the antibody and the red cells, resulting in hemolysis. Okay, so that's what it looks like under the microscope. Okay. Hemolysis is a lot of RBC bursting. Okay, that's what lysis means, right? To burst. And that's dangerous because the reason why someone might need to have a blood transfusion is because they don't have enough red cells in them, right? And that's why they need to have a transfusion to boost their red cell numbers. But if you're doing the wrong transfusion, all those extra red cells coming in will now all experience hemolysis, which would make the situation, not better and potentially worse. Right? So that's why we need to match the blood type before we go ahead with the transfusion. Okay. All right, we're gonna go ahead and talk about the RH blood group and then the pregnancy situation. That will take a little bit of time and quite a lot of focus, right? So please pay attention. Okay, so this is RH blood group. We learn about the ABO blood group and how the compatibility works, right? And you know, if someone has type A blood, it means they have the type A protein on them. And so now we shift our focus to another type of blood group called RH blood group. Okay, so the RH blood group is named after these monkeys. They're called rhesus monkey because the antigen was first discovered in them. Okay, so the RH blood group is referring to the presence or absence of RH protein on RBC. Do you have it or do you not have it? RBC with RH proteins makes the person RH positive. And then the next one is without RH protein, then the person is RH negative. Genetics of RH. There are only two alleles for RH blood group. The positive allele and negative allele. Okay, this could be a little bit confusing. Pay attention. Positive. If a person inherits two copies of the positive alleles or positive negative, just one copy of the positive alleles, both of these will result in RH positive. Okay, because the positive allele will allow you to make the RH protein. So it doesn't matter if you have two copies of it or just one copy of it. If you can make it, you can make it. Then you will be RH positive. Negative is the other possible allele. And that is going to make you RH negative. Okay, so like negative means you cannot make the RH protein, right? If both of them are negative, then you can't make any. So you'll be RH negative. So let's learn how to draw some of these RBC. Drawing RBCs, red blood cells. So let's say we want to draw someone who is A positive. A positive. What does a red cell that's A positive look like? Remember, we are talking about two blood groups here, the ABO blood group and the RH blood group. So if someone is A positive, the surface would have type A antigen, type A protein, as well as RH. Okay. What about AB positive? AB positive. What antigens will be present on the surface of the red cell?
A, B, and RH.
That's right. Thank you. A, B, and RH. Now, just to contrast this with a negative version, A negative and AB negative. In this case, we would have just A, right, with no RH. And over here, we would have A and B, but no RH. Okay, hopefully that makes sense. RH compatibility. Only negative can be given to positive and not the other way around. Okay. And it's easy to understand because people who are Rh negative, they don't have the Rh protein on them. And so they will be able to make anti-Rh antibodies, right? Individuals who are Rh negative can make anti-Rh antibodies, which would attack Rh positive red blood cells. That's why you cannot give positive blood to negative people. Positive can give to other positive, negative can give to other negatives, but only negative can be given to positive, not the other way around. All right. Okay, so the transfusion chart goes something like this, right? The same one as before. O can be given to A and then to AB like that. And then negative, Rh negative, can be given to Rh positive. So you must consider both of them when you determine compatibility. So let's say we have A positive to AB negative. Safe or not safe? Right. Let's take a quick poll and see what you think. A for safe, B for not safe. Okay, we have a lot of people who say it is not safe. Let's see what happens. So let's take a look at the ABO system, right? Can A be given to AB? If we take a look here, A is okay to give to AB, no problem. But what about positive to negative? Can you give positive blood to negative people? The answer is no, right? Because I just told you so. So if one of them is not okay, then this is not safe. All right. Let's go to our study guide here. And there's a question right here. Question number 8. And we have a bunch of donors here on the side, and then we have a bunch of recipient. So I want you to take a moment and fill it in. Okay, see which one is safe, which one is not safe. Give you 3 minutes to do this, and we will take it up. Meanwhile, if you have any questions, please ask. Okay. So the only ones that are safe are all the ones here. Safe, 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 safe. And this one's also safe. Everything else is not safe. Does anyone want me to elaborate on any one of these to explain further why it is safe or not safe? Any particular one that doesn't make sense to you? I can explain it to you. No? Everything makes sense? Okay, I certainly hope so. So please work on these study guide questions. This is another compatibility chart, right? It's good to think about who you can receive from and who you can donate to. You know, I'll post the answers later for you to compare it to, but you know, it's important for you to go through them. Yeah, just do as much as possible. There is another pedigree here. We'll do some of these baby matching questions. You can try them, but I'll show you how to do it properly next class. We still have quite a bit to talk about for the rest of lecture 2. We'll see if we can finish. Next Monday, I might have to leave early to go to a meeting. I'm waiting for the confirmation from the other department. So we might need to end like at around 10. For that reason, maybe also print lecture 3 for Wednesday. If we do finish a little bit early on Wednesday, we'll start on lecture 3. Okay, just to cover up a little bit of lost time for next week. Okay, there's one question in the message. Let me just read it. If only negative can be given to positive, but Rh negative have antibodies, so will it attack? Okay. This is a very common question for people to, you know, all of a sudden realize. Okay? The question is this. Rh negative can be given to Rh positive, right? But then I was saying earlier, Rh negative people can make anti-Rh antibodies, right? So here's the confusion that some people might realize all of a sudden. If you go ahead with this transfusion, wouldn't the antibodies then attack the Rh positive person's cells, right? The answer is no, because when we do blood transfusion, we do not donate the whole blood, right? We do not transfuse the whole blood. This is the exact reason why we have to separate the blood into RBC and plasma. Okay, and then you transfuse the RBC separately from the plasma, because all the antibodies will be in the plasma. So for a person who's Rh negative, when you do the transfusion for the red cell, you are not transfusing any of the antibodies. All those things will remain in the plasma, and that will be following a separate set of rules, right? I said. So that's why it's not going to be a problem. Does that make sense? Okay, so in RBC confusion, in RBC transfusion, only the red cell goes over, right? None of your antibody will be transfused. That's why you only look at the recipient's antibodies to determine whether it's compatible or not. So that was a good question. Any other questions? I mean, you can always email me, right? Okay. If you thought of anything. But that's it for now. I will see you all on Wednesday, 8:30. Have a good day. Bye for now.